大部分是脑科学研究所的学生，所以在座多少？
是大约。
好，因为我们其实人很少，其实不需要用太多大班上这个东西。那我们今天要讲的是在 digital medicine 这边比较偏向 precision medicine 的部分。那我想这个主题前几堂应该至少前2堂，谢老师应该跟我们聊过一些。那我们今天的角度会更偏向生物资讯这一块，所以我们要介绍所谓的 Omics。那 Omics 根据各位的背景，可能有些听过，有些没有听过。不过它很会发生在大家的临床医疗的健康生活上面。所以我们今天要看这个。那我们会用蛮多的影片来让大家认识这一块，因为这其实是相对新的东西。然后最近其实是我们的 YouTube 可以让我们有非常多的 video 用影片的方式来传达各式各样的概念，所以我们会用很多影片来让大家看这件事情。好，那第一件事情其实尤其是前2年 AI 啊，所以医疗这种东西还是讲的太多的了。不管去走趟小七全家，它上面的杂志应该或多或少都有一些 AI 相关的，USB 等等。那很合理的要看 AI 这块，医学上面、医疗上面也是一个很大的利基在里面。就像这边看到的，其实当初都会说，有些 buzzword 最近几年都出现了，什么大数据啊、资料科学啊、AI 啊。那当初也会有人觉得，AI 是要取代很多工作。那也有一派高的观点是说，其实就像我们以前的工业革命、资讯革命一样，有新的东西出来了，会减少某些人力的需求，但其实会创造其他的工作。所以随着最近几年的这些发展，比较或者是通过这个像是所谓的数据方面的资料科学家、资料分析师啊，其实都很高的。可能大家或多或少都听过以前，可能大概几年前，我在说所谓的资料流行时代的黑金可以取代石油。那我们现在市值最高的这些科技公司都是 based on data， 像 Google， 还有 Apple， 其实它最有价值的是 iTunes 这种各式各样生物的资料。那在健康上面呢，医疗和健康上面呢，可以想象一定会有很多可以导入所谓的 AI 的这些技术进去。但其实这不会是东西的，这不会是东西的，这从过去10年、20年就一直有人在做，只是还没有跟着 AI 这个风起飞而已。就像刚才有个书面说，你把猪放在猪洞口，它就会飞。所以这边讲的大数据和机器学习技术不是什么新东西，特别是放在药物筛选的设计，也一样不是什么新东西。那现在有的新技术，比如说现在的生物学习，可以在某些层面上面帮助克服以前没有办法克服的一些困难，或者整体概念上我们用电脑辅助、药物筛选，还有在我们的医疗决策辅助上面，以前就一直有在做。那现在的趋势呢，就是因为我们data资料累积越来越多，所以我们可以开始看各式各样的，不管是我们在医院里面的，还有我们跟一些开放数据，比如说大家市政府已经开放了一些资料，还有我们的中央政府开放的，比如说气象，还有各式各样的空污啊，环境各式各样的资料，还有交通。所以这些东西都可以结合。那这边的一个 perspective 就是觉得，我们未来会可以整合这些资料，帮助我们在医疗这个层面上面有突破性的发展。那像我的医生阅读啊，其实因为就是美国那边也在做这种东西，所以其实有学术界很多做这一块的，也开始有发表一些看法。比如说 data science 还有比较传统的生物统计啊、生物资讯，其实都在现在这个领域或者现在这个趋势上面扮演的技术的角色。那我们看一下这边的开头还是很杂志或是网络新闻的开头。不过呢，我要带大家看的是，谈到资料里面会是想到所谓的 database 数据。那我 一个海卵后的样子，其实就是大家很常见的我们的主母和我们的家庭成员之间的关系嘛。所以这张图在讲的事情，就是下面在讲的，我们已经可以对我们一个家族、一个人、一个家族，或是几百万的 population based 一个规模去做所谓晶体定序。那晶体定序，大家应该高中都是3类的，应该都学过生物嘛，所以基因的概念大家应该都 OK。大家应该知道人生体是基因 DNA。在我们最近大概20年，可能是我们开始有能力对我们的基因体进行定序，而且是用很大规模、快速的方式去做这件事情。那边的科技有那边的科技发展，但后端软体的部分、资料分析的部分，最近10年慢慢跟上了。像是我们常见的一些大公司 IBM、Amazon、Google 和 Microsoft。那有人可以告诉我们这几家大公司为什么会特别列的这几家公司？其实也跟最近的趋势有关系。有概念，不错。那这边列的其实就是我们目前市场上最大的云端运算的 player， 包括了 IBM、Amazon、Google 和 Microsoft。那目前应该还是 Amazon 的cloud市占率最高。那 Google 和 Microsoft 也慢慢追起来，目前 Microsoft 的市场还不错。那 IBM 是以前的大家都知道的通用运算机器，以前那个年代的电脑巨人。那现在它已经慢慢淡出了，至少个人电脑方面。那企业电脑它现在比较偏向做服务端 service 端，所以大家一样是做高computing，但是 IBM 提供的是比较 service 性质的。solution 性质的，那我们等一下可以看一下他这边提供给我们的，他们的角度来预测未来的，这边特别是讲 cancer treatment， 所以你可能会觉得很意外，为什么 IBM 会跟 cancer treatment 有关系？那可能有在看化妆杂志的，可能会知道所谓的 Watson，IBM 以前有一个，现在也还在发展的，算是人工智慧的系统，相当早期在做，所以相当早期的技术其实跟现在又不太一样。他们依靠相当早期的技术，大概知道他们一些事迹，西洋棋赢过世界冠军，还有参加了益智这种节目也是赢得人类的玩家。那最近当然是有试着在推动到医疗上。那如果最近，特别是最近，大家有去一些国内的研讨会啊，或者是相关的会议，那你可能会听到有些采用了 IBM Watson 的医院，在抱怨好像也没有那么神奇。Watson 系统对他们帮助是有限，然后它的帮助可能是在天下杂志啊，或者什么杂志可以帮他们写个专栏，让大家以后看到了什么 AI 觉得很厉害。不过你其实到我们就是巷子内的研讨会去看的话，你会发现其实它还是有很多限制的。那我们以后可以再慢慢看它的优缺点在哪边。不过我们今天重点是在讲这些云端平台它提供的资料分析服务，目前绝大多数是可以利用人类资料。所以目前在讲 personalized medicine 或者 precision medicine， 绝大部分都是从这个层面去看。我们今天提供了我们什么资讯，让我们的临床医疗做出的决策跟和以前有什么不一样？好，那我们很快的用1分半的时间看一下 IBM 所提出来的，他们认为未来的科技是怎样去帮助我们做 personalized cancer treatment。那这个影片其实是大概5年前的影片，里面的一些愿景其实某程度上已经实现了，还没有到全面的规模。那可能看一下英文字幕，让大家可以比较看有没有跟上。好，开始了，1分半，所以很快。
So do you want the good news first or the better news? First, the good. So do you want the good news first, or the better news? First, the good. In 5 years, cancer will be treated down to a DNA level with treatment plans that will be more personalized and accurate. With growing aging populations and global cancer rates expected to jump 75% by the year 2030, DNA-specific treatment is good news indeed. Today, crunching the patient's full DNA and formulating the targeted treatment plan can take many months if a patient is lucky enough to have access to the right medical team. But within 5 years, sequencing DNA could take only a day and become the standard of care. Cloud-based cognitive systems will crunch reams of medical information, helping doctors give an individualized treatment plan in just days or even minutes. This time saver could be literally a lifesaver. Oh, and the better news? These systems in the future could recommend to doctors DNA-specific treatments for conditions like stroke and heart disease. Through the cloud, smarter healthcare will provide access to more information in more locations using fewer resources. And our learning systems will keep learning to get smarter and smarter with time.
所以里面谈到的一些东西啊，就是我们怎样，他们的愿景是以后我们可以怎样用刚刚说过因为资讯帮助我们去做什么客制化的医疗服务。personalized cancer treatment 都是在那边讲到 cancer treatment， 它的确是用你可以说精准医疗这个词的应用最成熟的领域。那我们等一下可以再看一些例子。那我们现在先回到投影片。好，那他刚刚提到，其实除了cancer treatment，他也提到了也有 stroke， 比如说我们团队就可以在跟学校其他的 stroke 研究中风的团队一样去利用我们的资料库做一些中风方面的研究。那刚刚提到的一件事情就精准医疗。那刚刚里面是提到的 personalized medicine， 那其实跟 precision medicine 你可以把它当成某程度上的同义词。那这个词为什么会这么红，或者是为什么现在世界各国在往这个方向走？当然也是因为美国他们有意愿的朝这个方向有进行一个国家型的计划。所以大概在几年前，奥巴马在任内的时候就宣示 precision medicine 是他们国家的计划。那这中间到底是什么？其实你看图就知道了。刻意把一个像花生配当初在普通会上的时候秀出来的一个东西。也就是说，我们可以开始用分子生物的层次 ，DNA 层次的资料还有它的资讯，帮助我们改变一下我们在很多医疗决策上面的跟以往不太一样的方式。那最重要的当然就是经济地区的，我比较强调的是经济地区的，然后透过我们结合我们其他的临床资料库以及健保资料，大家都知道我们台湾有个很强的健保资料库。在英国或是有国家型健保的地方，其实这资料相对完整。美国虽然没有国家型的健保 ，Obamacare 算，不过他们的民营的健保、民营的健康保险的业者，他们的一些资料也相当庞大的。所以，如果大家以后有兴趣去看相关的研究的话，你会看到一些先进国家很多利用这种保险资料库啊、相关的资料库做研究。好，那我们因为这有它历史上面的重要意义呢，所以我们可以看一下当初白宫所秀出来的宣传影片，一样1分44秒，快。好。
Precision medicine is an exciting new area of medicine that allows us to use cutting-edge technology to match patients with the treatments that will make them well. Precision medicine gives us the tools to say what makes each of us different from everyone else. And that's a mixture of our genes, the microbes that live on us and in us, the environments that we live in, and all the factors in the environment that we're exposed to, as well as our diets. All of those things come together to determine how healthy or unhealthy we are. And the power of precision medicine is beginning to interpret what factors will affect an individual's health or susceptibility to disease. We have technology now that can sequence the human genome quite rapidly. That means we can get a sequence or the alphabet that forms the gene for every single trait in the human body. Precision medicine is now. We're already using it. It's already affecting people's lives, and we know it works. But if we can expand it to cover lots of different disease and health characteristics, we will be able to treat all sorts of diseases that we have no idea how to tackle right now. So the future of precision medicine is enormous. This is a revolution in medicine. I am so excited to be here at the White House working on this initiative because I think this is one of the biggest things to happen in medicine in a very, very long time. And will really make the future health of the people of America totally different.
So precision medicine precision 到底是什么意思？为什么可以称作精准？那这主要之前有一些解释呢，它跟 personal medicine 某程度上是同义的。personalize 什么意思呢？个人化、客制化。那为什么需要说个人化、客制化？是因为大家的天生得到的 DNA 序列或多或少都有一点不同。那这有以前称为体质的东西，我们现在可以用比较科学的方式去描述。那另外就环境因子，大家的成长、我们的生存环境、我们的工作环境，各式各样的都会造成各式各样不同的影响。有先天的影响，后天的影响。Together 我们需要掌握这些东西，才可以帮助我们更好的客制化，对每一个人最适合他的医疗策略啊，或者照顾策略。所以理想上是这样，不过大家也可以发现，其实我们现在理论上还没有做到这种境界。不过我们对于 DNA 基因体的了解的程度，已经算是这个科技已经就位了。那现在在等的就是商业化、普及化，各种法规、各式各样的东西跟上来。所以你可以想象，这种东西最一开始的受益对象是什么？有钱人。他们才有钱去负担的其中，大规模的基因检测啊，那检测出来如果需要做去，可能大家也听过现在的标靶治疗，或是对他的免疫治疗、细胞治疗，都健保不会给付的，因为它实在很贵，而且还没有办法保证它的效果是，可以普遍适用到所有 case。所以普及化是目前大家可以等待的。一个可能在最近5年、10年之内会慢慢普及化。那我们可以看一下，在普及化之前走得比较快的一定是学术界，不管是我们的医学院的大专院校，比如说我们，或者是比较先进的医疗机构，他们经常都会 sense 到 precision medicine 以后要怎么样去 shaping 后的医疗的面貌。所以我们可以看一下，比如说这个，我是觉得不错的，它来自 UCSF。大家都知道 UCSF 的医学院是世界上的名，因为在美国也是首屈一指的。所以我们来看一下，一样是1分50秒就解决，是短短的。
Precision medicine. You've probably heard the words on the news or even at your doctor's office, but how does it affect your life right now? Precision medicine is basically a modern approach to understanding health. Take asthma. Asthma affects more than 25 million Americans, nearly 1 in 12. They all have one thing in common, a condition that makes breathing difficult. In most clinics, these patients are treated with an inhaler and the same drug, albuterol. But are 25 million people all the same? If you have asthma, your condition depends on where you grew up, how old you are, and your ancestry. It also depends on the air you breathe and if you have carpets or pets. Every patient is unique, so one-size-fits-all remedies don't always work. It's not just asthma. For diabetes, cancer, and most other diseases, today's treatments are based on obvious symptoms, standard tests, and common drugs. Precision medicine, however, starts with creating a clear picture of health by collecting, connecting, and analyzing many kinds of information. This could be data you provide from your fitness tracker, or it could be what scientists are learning in their labs right now about the underlying causes of disease. Genetics, your health history, and other details are added to the mix. When we integrate such data from millions of people, your clinician and researchers can understand your health risks, how others like you responded to a disease or drug, ways to prevent disease, and what makes you unique. So we can stop focusing on diseases and care for you as an individual. The more information we collect and connect, the better scientists will understand health. And as that knowledge network grows, we can ensure that every patient everywhere gets the right treatment in the right dose at the right time.
好，那其实他讲了很多刚刚提到的面向，那我们可以再回头 summarize 一下。首先呢，当然是每一个人，就像刚刚说的，他的因为去上述过程都不一样。那同样的 treatment 不是对于每个人都有相同的效果啦。那传统上呢，我觉得我们现今的医疗是这样做 ，common symptoms 我们就给他 common 的 test 或者是 drug， 所以其实是一个 symptom based 的治疗策略。那包括癌症治疗也是啊，如果你是得了同一种癌症，然后在同一个阶段，通常就有标准流程下面的同一种treatment的东西。但现在也不一样了。那如果大家有了解这一块的话，我们现在可以慢慢做到说基因检测，表面上的肿瘤上面的biomarker 检测，可以帮助我们去知道要用哪一种treatment对这个患者最有效。那后面他当然就提到了我们需要做的是什么，当然就是要收集各式各样的资料，包括人类的资料，还有现在已经很红的，就是其实很多新创在做这一块，包括 Apple， 你可以发现他们的策略也是在给你各式各样穿戴装置来收集的资料。So all about the data， 那包括我们的 DNA 序列、遗传方面序列，还有我们的用药的结果、饮食习惯，那这些东西都是配合我们政府的资料库、用药的保险，还有现在台湾也在推健康存折。所以你的医疗记录都是在中央政府下面是有记录的。那这一块当然是大家都想要知道的资讯。那目前还有法规上还有伦理上面的没有厘清，所以目前可以应用的相当有限。比如说我们现在用健康存折上去，你其实插个健保卡，然后你拿自己可以去申请一个凭证，然后去看你自己的健康存折。大家可以看到过去3年你的就医记录。那更之前的，大家可以追寻到你的疫苗接种记录。那现在你这一代的疫苗接种应该都有电脑进档。我那个时候疫苗接种只有给你一张纸本的卡，俗称的小黄卡，你可以回去问你的父母。那这有什么不方便的？比如说当初要出国留学或工作的时候，你的机构会要求你要打一些疫苗。我的意思卡是你要什么牌号？是这样的。那理论上你打过了，你只要带那个记录过去，他们就会跟你说不用打，只要去附加一些自己需要失效的答案好了。但以前的纸本记录，其实卫生所不一定会留。那就算你自己有留下小黄卡，你还是需要去卫生所给你一个凭证。那我自己的经验是，我当初碰到的经验是，我带了小黄卡去卫生所，但卫生所不发给我英文的凭证，因为他们这不是他们常见的业务，所以你们可以想象，有些卫生所就不愿意做这件事情。那后来怎么解决？其实基本上有2种解决方式，一种就乖乖去国外，再全部都打字；另外一种其实就是你去找医院嘛，跟卫生所施压一下，医院开给你。所以我后来是取第2种。这有点悲哀的，其实你会知道有很多政策在推的，这个课后就不要听了。很多政策在推的时候，最大的阻力现在是政府的公单位，特别是比较传统的公务员，他们觉得其实是多一事不如少一事的。好， 这有点离题了。不过我们这边的愿景，像刚刚说到，你可以想各式各样的资料。那接下来呢，我想大概就讲到这边这块了。
Should we collect and connect the better?
所以为什么我们数位力学这门课要讲到 Precision Medicine？ 因为它其实说穿了背后都有一大堆资料。你可以看到各式各样的所谓的 heterogeneous 一起性的资料，然后这样子合在一起。然后更困难的是，这些资料不是一个时间点而已哦，它是你的过往的 history， 过去的生活就业的轨迹，所以它是有个时间轴的。然后怎样分析这样的资料，其实一直是有新的研究、有新的方法出现的。好，我们回到本片。来，那一切当然不是这么美好了。其实就像当初我妈妈在说我们要国家我要推Medicine的时候，当然有一些concern出现。那大家可以想象一下，有什么可能的问题？有什么可能的问题存在？所以那个时候，当然其实就有做一篇比较短的介绍说，Medicine 算是什么？实际推动上面会有什么问题？所以你会看到他访问是比如说CUA 是一个公标的 Medical Center 的这些参与 Medicine 的人。那我觉得我们可能4分钟的时间也 OK 啊，花4分钟了解一下 Medicine 有什么坏处，不要是这样好的，通常要讲一下它的问题。那我们一样看一下这个案的记忆。好。
This is going to be completely unprecedented. It is going to affect medicine in ways we've never seen before.
You can imagine blood transfusion to a blood type. What if matching a cancer cure to our genetic code was just as easy, just as standard? And that's the promise of precision medicine. Delivering the right treatments at the right time, every time, to the right person.
Precision medicine enables the ultimate delivery of personalized medicine, which means that you come to your doctor, you have your genetic information, and the doctor can use that genetic information to specifically design treatments for you. But obviously, there will be thousands of individuals that have your disease and your gene. And so it's personal for you, but it's also treating large numbers of people in the same situation. We can look at individual tumors. We can even look at individual cells within tumors and understand how their system is going to arrive. It's possible to analyze this very complex data using state-of-the-art computer algorithms to be able to see the interaction of the proteins in the cancer cell and to design drugs that will inhibit or treat them. A really good example of how all this works is with a drug called Gleevec. And this is a drug for a certain type of leukemia. This drug had a miraculous effect. I mean, it basically cured the disease. However, the individuals will receive this 6 months, a year later, it was back. Then a second drug was developed. We had the tools to go back, look at the sequence of that reverted protein and know where the change was, and then design a second drug to address that. And now the combination of those two drugs that are used in sequence basically has cured one form of this disease. And so I think that's what's going to happen. There'll be more and more examples of cases where we can join genetics, biology, and medicine. In heart disease, the chairman of physiology, Andy Marks, has been studying a receptor that is absolutely central to how the heart functions. And recently, they determined the three-dimensional structure of this protein. That has then led very quickly to the development of small molecule drugs that address certain heart disease. That defines precision medicine because it can't be more precise than having a high-resolution three-dimensional structure that tells you something about the human disease.
What's so exciting is that we have the possibility of leading an entirely new era of medicine that makes sure new jobs and new industries and new life-saving treatments for diseases are created right here in the United States.
5 years ago, it was about $10,000 to sequence a whole genome. 10 years before that, almost $1 billion was spent worldwide on sequencing this. Today, we can sequence a whole genome in 24 hours for $1,500. But when you take 1,500 times the number of people you need to see before you begin to learn the relationship between the genetics and medicine, that's a very big number.
It is difficult to forecast exactly what the financial implications of precision medicine are going to be. On the one hand, if drugs are tailored to very specific populations, the prices are higher because the value to the patient is also higher. On the other hand, today, many people are taking drugs that are really not doing them any good. We can avoid the unnecessary costs of inappropriate medication. That would be cost saving.
It's important for the lay public to understand that this doesn't happen overnight. The genetics is now straightforward. We can sequence very rapidly, very cheaply, but we still have to add on to that the drugs developing, and that takes time.
OK， 所以看完大家知道他讲的其中一个问题，就是随着越来越 precision， 越来越 personalized， 那其实是不是就落到跟罕见疾病的 treatment 一样的问题？你的 target population 就是这么小，然后没有个药厂愿意用那种经济成本去开发新的药，或者是这些药非常贵。那另一方面 ，precision medicine 但也要跑出像他刚刚说的，现在有很多药是不该开，开了都没效，但是按照 SOP 就是必须开。那未来或许可以 follow 这样的指数呢，所以有带来一些好处，但也有一些未知的隐忧，所以这是我们未来应该要知道的事情。那其实它里面有讲很多的，我们帮同学去看一下。好，那他讲的precision medicine， 其实在前半段有些 information 可以再解释一下。好，比如说这里我们看当然可以结合很多的医疗影像。那在分子生物的层次，我们也可以去看，就像他说的，我们可以看到肿瘤内部的组成。我们现在必须的解析度可以达到所谓的细胞层次。那我们知道一块肿瘤里面不会单单只有一种细胞。那现在在讲 cancer 的时候，越来越多是讲所谓的 microenvironment， 它整个微环境。所以那个癌细胞要能存活，不仅仅是他一个人 OK， 他还要依靠他周遭的细胞，提供他的营养啊，提供他可以转移的空间啊，所以像目前这一块研究所做的多发展的，我们怎样去用单细胞的确的技术，把每个肿瘤里面的异质性搞懂。那后面也提到，我们可以再去看，比如说这边是有提到的，我们需要资讯方面的帮助，比如说 state of the art， 然后 algorithm， 所谓演算法如何去从分子层次的资料得到我们刚刚想要得到一些 information。那比如说 interaction of proteins 是很重要的东西，我们知道蛋白质和蛋白质之间会互相相互作用。那传统上我们只能用很简单的实验，一次去抓2个蛋白质下来去看相互作用。但我们在细胞里面，活体细胞里面，在生物生理现象、生物功能的时候，其实很多蛋白质之间的交互作用。所以要怎样去做出一些模型来预测一些蛋白质交互作用，也是这一块在做的事情。那后面的话，应该都提过了，我不再讲了。那我们就回头来看同一篇。好，那刚刚讲的东西，其实已经大家把这整个概念讲清楚。那我们这门课大概是希望给大家一些更细节的东西，因为大家都是在这一块，这一块生理学。 家都是未来会对这一块的了解更透彻一点，所以我们要利用这个机会让大家看一下所谓的 sequence 到底是什么。我们要去定序所谓的 human genome， 它到底是一个怎么样的概念、怎么样的做法？那它其实就是我们所谓目前在谈的 precision medicine 的基础啊，基础中的基础。所以我们花一点点时间，用这个 TED education 的影片，只要5分钟，觉得很好，可以让大家很快的抓到 sequencing 我们的 human genome 是什么意思。
Grammarly does more than catch errors. With Grammarly, you can find
You've probably heard of the human genome, the huge collection of genes inside each and every one of your cells. You probably also know that we've sequenced the human genome. But what does that actually mean? How do you sequence someone's genome? Let's back up a bit. What is a genome? Well, a genome is all the genes, plus some extra, that make up an organism. Genes are made up of DNA, and DNA is made up of long paired strands of A's, T's, C's, and G's. Your genome is the code that your cells use to know how to behave. Cells interacting together make tissues. Tissues cooperating with each other make organs. Organs cooperating with each other make an organism. You. So, you are who you are in large part because of your genome. The first human genome was sequenced 10 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries, and cost over $3 billion. But someday, very soon, it will be possible to know the sequence of letters that make up your own personal genome, all in a matter of minutes, and for less than the cost of a pretty nice birthday present. How is that possible? Let's take a closer look. Knowing the sequence of the billions of letters that make up your genome is the goal of genome sequencing. A genome is both really, really big and very, very small. The individual letters of DNA, the A's, T's, G's, and C's, are only 8 or 10 atoms wide, and they're all packed together into a clump, like a ball of yarn. So, to get all that information out of that tiny space, scientists first have to break the long string of DNA down into smaller pieces. Each of these pieces is then separated in space and sequenced individually. But how? It's helpful to remember that DNA binds to other DNA if the sequences are the exact opposite of each other. A's bind to T's, and T's bind to A's. G's bind to C's, and C's to G's. If the A, T, G, C sequence of 2 pieces of DNA are exact opposites, they stick together. Because the genome pieces are so very small, we need some way to increase the signal we can detect from each of the individual letters. In the most common method, scientists use enzymes to make thousands of copies of each genome piece. So we now have thousands of replicas of each of the genome pieces, all with the same sequence of A's, T's, G's, and C's. But we have to read them all somehow. To do this, we need to make a batch of special letters, each with a distinct color. A mixture of these special colored letters and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome. This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book which you don't speak. You can recognize all the letters,
but still have no idea what's going on. So the next step is to decipher what the sequence means, how your genome and my genome are different. Interpreting the genes of the genome is the part scientists are still working on. While not every difference is consequential, the sum of these differences is responsible for differences in how we look, what we like, how we act, and even how likely we are to get sick or respond to specific medicines. Better understanding of how disparities between our genomes account for these differences is sure to change the way we think, not only about how doctors treat their patients, but also how we treat each other.
好，那我喜欢这影片的原因是它把目前的定序技术用很简单的方式讲完了。那所以大家可以想象吗？我们要定序一个 DNA 的序列，不是从头读到尾。你的 DNA 序列有多少？3乘10的90方，所以不是像我们读书一样从第一个字母可以读到最后第3乘10的90方的字母。那他们用的策略其实相当聪明的。那我们回到前面吧，回到前面，我们回到这一块。那我们用的策略就像他们说的，所谓的 shotgun sequencing。那为什么叫 shotgun？ 散弹枪。先把 DNA 很长一条，把身体的 DNA 抽出来，打断，然后把它们丢到一个平面上面，它就会结合到我们比如说不会载片或是各式各样载片上面。所以每一段都是小小的一个 DNA 序列，然后试着让它去放大增值。这一步很重要。因为如果只单一一个片段，它释放出来的阳光信号通常不够我们侦测的支持度，所以我们需要用 PCR 放大，把这些片段放大成一个 cluster， 就像大家这边看到的这样的一个 cluster 里面的片段是一样的，那我们就可以用他刚刚讲到的方法，去让它结合各式各样的带有光的酶，碱基啊，带有光碱基会发亮。我们就可以去读出来一个序列，所以它修出来其实就是目前最常用的所谓定序。那像一台大概要平的要几百万，那我们基因体中心有几台？那比较有趣的，它其实也有出比较小台的，便宜一点，那你可以放在实验室里。那如果你够有钱，你也可以买一台放在家里，想要定序其实就定序。那这里也不是很可笑的事情，因为还蛮多地方现在开始在这样做。那有另外一个领域叫做Omics，它其实就是你空气里面、水里面，或是你的大便里面，去抓出这些素里面的 DNA， 那你就可以知道你这个环境里面，你的水、你的空气、你的大便里面到底有哪些生物，通过 DNA 定序。那这种东西如果做成 Omics 可稀释的话，你可以想象它的威力了。可以在各式各样的不同的环境，比如说雨林啊、反对期啊，各种环境下面去采样，得到它那个地方的物种的丰富度，所以有各式各样的领域可以用。那后面再是讲这边了。所以我们刚刚讲的 medicine 某程度上面就跟我们在看一些比较所谓的模型是一样的。那我们都知道从大 A、小 A、大 B、小 B 其实都以传觉方法去看我们的 genotype 和 phenotype 基因型和表现形之间的关联。那我们现在定序可以找出来哪一个碱基的什么样的改变可以连接到什么样的表现型，比如说连接到你的发射、左手和右手。那有些东西当然不是这么单纯的，不是很简单的说哦，你这个基因有这个碱基的字母就会对到那个phenotype。大部分情况下，我们只能找出一群碱基 ，OK， 因为序列上面的碱基一群，然后各自跟这个表现型的关联有多强 ，OK。那这种研究通常叫做 genome-wide association study， 就是从整个基因体上面去找所谓的 association。phenotype 之间的 association。那我们很多目前的医学研究也是based on这个方面，这种研究策略去做的。然后后面提到的当然就是我们genome的研究怎样去帮助我们，然后提到就是 medicine 这一块概念。好， 一节课可能剩下1分钟，看我们可能就跳过大部分的东西。最后讲一下这个吧，既然这一门课有带到Biomarkers，所以要看一下Biomarkers。大家认识他，讲这些事情要找他，一定要注意。那他在大概2013年左右就有很大的事情，就是他做了乳房切除术，就是因为他的医生就建议他进行这个，因为他有家族的乳癌的历史，那他有做 genetic 的 testing， 他知道他的 BRCA 这个肿瘤抑制基因有突变，所以有这个突变会造成妇女得到乳癌的机会有显著上升，像是我们下一页看到的。BRCA1 和 BRCA2 这两个基因呢，它会做出什么抑制蛋白？它可以做什么 DNA 修复？或这两个受损呢，你其实细胞上会接受一些DNA 上面的破损，或者是一些 mutation。那有肿瘤抑制基因帮我们修复的话，就可以避免一些细胞突变产的情况啦。简单来说是这样，所以有这些突变的话，会造成蛮高的几率。有可能会发展成乳癌，特别是你有家族病史的时候。所以我们可以看到有 mutated 的突变的BRCA1到乳癌的几率其实变得非常高的。那如果是正常的BRCA1、BRCA2的话，其实只有10%。所以这其实就有点在预防医学的层面上面，我们做一些基因检测，那你是不是有任何的处置可以帮助我们去预防，就是那个病去。那时间到了，那我剩下1分钟，我想跟大家讲一下，其实这个事情已经发生了蛮大的效应了，也就 Time Magazine 他当初会用这个字的原因，就是呢 effect。我们第一次看到有个名人愿意做基因检测，还没有发生乳癌之前，愿意就这个几率、这个风险去做一个侵入性的 intervention， 乳房切除。所以这件事情带来的影响是，随后的5年到现在，英美国家的妇女接受基因检测包括BRCA2的比例比以前上升了非常多。那乳房切除术上升了不多，但大家至少愿意去做genetic test，这就是 effect。好，我们有2节课嘛，那我们第1节课应该就讲到这边，那我们下一节课会开一下 Omics 是什么东西？所以我们一样下课10分钟，我们抽手再回上课，谢谢。那我们上半节其实上一节precision medicine， 特别是在生物资讯小组讲的比较清楚的。那我们现在要花一点时间来讲所谓的 Omics。那看这个字，大家有发现什么东西？刚刚在讲的基因 Omics， 它的结尾就是说的 Omics。所以现在它单独出来变成一个字，它指的是什么？其实我们细胞里面除了基因体之外，还有其他的体。那就像这边讲的，还有所谓的 transcriptomics， 它这个东西什么 ？DNA 会做出 messenger RNA， 那这些 RNA 的总称就叫 transcriptomics。那这些messenger RNA 会再变成蛋白质的 protein。那我们细胞里面蛋白质的总称就叫 proteomics。那我们还有一些小分子代谢物在细胞里面。Metabolomics， 这些东西它的结尾都有这个后缀 omics， 所以它就变成一个独立的词了。讲的是什么呢？我们所有这些 information， 不管在 genome、proteome、metabolome， 各式各样的东西结合在一起，就是说 collective characterization and quantification， 所以这些分子我们都把它收集在一起，从里面得到各式各样的资讯。Together， 它其实就描述了我们细胞的生活动现象，到底形成哪些分子哪些分子之间交互作用。所以为什么这个重要？因为我们刚刚提到 precision medicine， 大家看到影片，大部分都在强调 genomics 这一块，因为它会发展的比较早，比较容易理解。不过未来呢是要整合所谓各式各样的所有知道的分子资讯一起丢进来。那包括刚刚所说的很多 treatment， 其实不仅仅是要从 genomics 上面看所谓的 genetic barrier， 我们基因上面的遗传的会比较部分，遗传的变异。我们还需要投入一些药物嘛？做一些treatment，它就会牵扯到这些 些医学杂志很流行看到这样的一个旁边的这样子，它的描述的就是我们从 B 到22加上我们的生产基地 XY， 它们之间在各种形式上面的变化，它的表现上的变化，它的 variance 的变化，都可以用这种视觉化描述。所以我们的 human cell 其实就像刚才我们的设定，其实是缠绕成一个像 turbo 一样的东西，打开来这最常见的一个形态，然后端粒图，还有上面有动画片，然后 DNA 上面还有一些讲机化的叙事，那这些都是会影响到我们DNA 怎么展现它的功能。那刚刚讲过的 metabolized proteins， 还有它可以转录出来的 RNA， 这些东西我们如果都可以把它搞清楚的话，就 Omics 所要做的事情。那同学可能会觉得很奇怪，就是画一个太空人，还有像是宇宙的地方，这边看起来也是 NASA， 所以这是一个 NASA 做的团队。那 NASA 为什么会对这个有兴趣呢？我每次在讲这个主题的时候，都喜欢用他们做的东西来跟大家讲。那也就是说，除了医疗领域之外，就连 NASA 中要上太空的，都对我们的分子生物学 Omics 有兴趣。为什么？那其实因为他们体验到，我们毕竟是要送人上太空，人上太空之后会有什么样的，它的细胞会有什么样的变化，跟我们在地球上有什么不一样？这些当然对他们来说至关重要，太空人至关重要，太空任务的成功与否也取决于你太空人在外面有没有健康。所以这是当初的一个计划，我觉得可能大家都有听过，因为南有是一个双生子的计划。他们去找双胞胎太空人，一个留在地球上，一个送到太空上，然后就等于是一个 case 做的实验，去比较一下上太空带来的影响，尤其是在 genetic 层面上面到底有什么不一样。所以我们可以用很快的时间看一下，其实有到5分钟时间看一下这个影片。那这影片之所以重要，是因为他们NASA去找了我们这个领域，特别是在做 Omics 的领域，可以说是首屈一指的一些来负责的计划，所以这个影片是很大的影响。所以大家可以看到，它其实有一系列的讲 Omics 这个面向，那我们可能会看其中的1-2个比较基础的部分。
I think we're all healthy most of the time, but there are times we get sick. Sometimes we get really, really sick, and the consequences can be quite serious.
Michael Snyder， 他最近朋友来台湾，其实是朋友来台湾，他还比较来台湾。最近可能是去中国跟他开始他吧。那大家可以看到他是 Stanford， 那他也是我这个 Twin Study 的 investigator。他找他来其实蛮合适的，因为他在这一块是深耕很久。
There are several problems with our health system. First, we actually treat people after they get sick. We actually don't do much to keep them healthy and understand what's going on while they're healthy. And the other is we're in a one-size-fits-all. We basically give people the same treatment, even though we're all individuals, and even though the way we may respond will be very different. So what I think we need to do is switch from this one-size-fits-all to something that's individualized, where every person is treated according to their genetics, all their life history, and their environmental exposures. There's an amazing revolution going on right now. Omics is really seeing the entire collection of molecules that make you. Basically, it's your DNA, it's your proteins, it's all your metabolites and such. The sequencing of the human genome was a huge, huge major step in making all this happen. So for the first time in 2001, people were able to decode the DNA of a human being. And that basically set up a reference onto which we could actually do a lot of the mapping we're talking about. We can now sequence put all those pieces together. That's the same for omics. You basically try to understand all the individual pieces so you can see the whole picture. NASA Twin Study is an amazing unique study where we're studying Scott and Mark Kelly. In this study, we have one twin launching into space undergoing incredible stress responses during launch and during the time he's living in space. And the other twin, the identical twin, is living on the ground a normal life. And really what's nice about Omics is we're going to see a picture of both of these astronauts at a level that's never been looked at before. We're going to be making billions of measurements, the one on the ground and the other one in space. And we'll see what space really does to the human body. I think this study will be very valuable for understanding space travel because ultimately we hope to send people to Mars. And that's going to be a long period, probably several years. By getting this IMAX big movie, what's going on is people see space radiation, space food, all the environmental impact of space. We'll be able to better treat, hopefully, astronauts and manage their health while they go on this incredible journey. Omics is really an amazing field where we can look at people and their health at a level that's never been possible before. And with that, we'll be able to better manage people's health and try and keep them healthy, again, long before they get sick.
好，简单的选择题，让大家看好之后，很快的 test 一下 Omics 到底是什么？答案是 B。OK，OK。好，那我们回来往篇。那接下来第一篇其实就可以再讲所谓的genome。那genome的部分。跳过去好了，因为我们分我们上一节课已经讲蛮多的，可以花点时间讲其他的这个。那这边我也大概讲一下，就好，不需要看。那我一直在强调 genome 很重要，我们 DNA 很重要，我们上面的传递也很重要。不过其实大家有留意的话，我们实际上也知道我们 DNA 上面的一些修饰，会影响到这些 DNA 怎么表现。然后有趣的是，这些修饰是可以透过环境做改变的，可以透过环境改变。有在看一些相关的新闻，前阵子有篇叫做“你老爸的打鼾声会影响到你下半辈子的健康”。那你可能在那个时期，然后你老爸可能睡在你旁边，他可能有打鼾声让你睡得很差。那像这样的环境压力，其实有在你的 DNA 上面留下一些讲实化修饰，可以说它是遗传上的印记 ，OK， 它会影响你的 DNA 表现。所以这边讲的是我们以前其实就已经知道了，我们要在 healthy 的 lifestyle 其实是可以帮助我们克服一些跟心理上面比较不适的东西。那这也就为什么现在除了强调 genetic 上面，要强调我们的 lifestyle 和环境。怎样去影响到我们的机体层面上的东西？比如说 PM2.5 就是像为什么一直在讲 PM2.5，除了它表面上的这个微粒一定会影响到人呼吸系统上，有很多相关研究会在讲噪音PM2.5上产生的冷，其实会在你的 DNA 上面留下一些算是 scar 啊，那这些都是透过讲话修饰还有各式各样的除了外遗传学上面的修饰影响到你。好，简单来说这样，超过这个来看。好，那 Omics 它刚刚讲的其实就是整合我们所有的分子嘛，我们 DNA 分子、我们RNA分子，我们有些蛋白质、代谢体，那这些东西就像这边所秀的，它在最底层上面其实是非常的 organistic， 那这代表什么意思？我们每一个生物它基因上面都有多少不一样，它做出来的那4秒钟metabolized 都多少不一样，这样很合理。那在比较高层次的情况呢，我们开始去厘清这些 DNA 彼此之间怎样调控，比如说这些蛋白质彼此之间怎样会去调控， 到毛皮肤，所以我们可以大概归纳出来这些基因调控的零组件长什么样子，就跟我们工程学上是很像。我们为什么会有这样的电脑？现在为什么有这种高手电脑？我们是先从一个简单的 free file 做出来的。然后这种我们忘记我开的正反器之类开始都出来了，所以会有正反器都出来电晶体，然后有晶体电路，然后到现在有所谓的 SOC 就 System on Chip。所以我们就是用这样很微小的零件组出来一个很功能很复杂的生物体，或是我们的电脑。这边也是一样的，我们透过这些微小的零件，我们可以组成一些有功能性的组件。那它可以参与到所谓代谢反应，还有所谓的调控的路径。那可以更高的层级去看这些东西组成的模组性、模组性。所以我们把这个 collection 再把它从更高层级去看，然后发现不同的模组它们之间是很 collection， 不同的配置位之间，但不可能就独立快乐，它跟其他的配置位之间有同程度上的联系。那我们可以再从更高层级去看到 scale 来看这些东西的组织性有什么样的性质。所以越下面代表就是我们的后面的 specificity， 翻译性越高。但是从越高层级去看的话，其实我们可以看到很多的生物体或者生物现象，很多细胞，它们在模组或是高层级组织体系看到的是 diversity， 大家 follow 的物理规则或者是生物物理现象是相当一致的。那好，这边大概就先讲到这边，看下一页。那 Omics 这个研究其实有，你可以说它有另外一个名字叫做 Systems Biology， 系统生物学。为什么？因为刚刚所说的各式各样的 Omics 基因体、传播体、蛋白质体，它其实都是把它们想象成各自的系统，都是一个系统。那我们在生物学、分子生物学这一块，就是因为之前的科技，让我们可以对各式各样层级的这些分子得到很多 information， 所以就有一个领域叫生物biology出来了。那其实这是目前生物学上面很大的主流生物这一块。比如说我们生物学上面会问一些生物学上的 question， 那就trigger了一些 technology 出现。那这个 technology trigger 也就刚刚所说的 DNA 的定序理嘛。DNA 定序理这科技出来之后，看我们的根据攻击码的，你就得到非常多的新的资料。那就需要很多 application 的人进来，他们需要开发一些新的软体，他们需要透过一些演算法或是什么统计方面研究，得到一些新的 process， 来 trigger 我们的生物学家得到 new insights 新的洞见，甚至 trigger 出下一个阶段的新的生物上面的 questions， 然后又引领到我们有新的 technology。就刚刚说的 DNA 定序也是经过好几代才有现在的状况，才有现在的可行性。那未来其实还有第3代、第4代的定序慢慢出来，它功能更强，它的成本更低。那这是一个很良性的，我们在做生物biology scientific discovery 的 cycle。那特别是我们做生物资讯的这一块，大家就其实到 information， 会去了解资料，甚至从资料里面得到一些新的开发式的 insight 或生物学家做验证。这个是一个良性的开发循环。好，那细节的部分我们不再讲太多，我的英文来看。那这是刚刚那个 NASA Omics 的短片。那我觉得你应该不需要特别讲的，可能刚刚提到的，大家都提到 epigenomics 有提到 EPI 是什么意思 ？epigenomics， 所以 genomics 大家都已经很熟了，就我们说的 DNA 的 collection， 加上一个 epi， 其实就我刚刚提到的你老爸的打呼是会影响到你下半辈子的幸福，这是因为它在你的 DNA 外面做的修饰。epigenomics 研究的就是我们所谓 genomics 外面的那些遗传信息，比如甲基化修饰啊，各式各样的修饰。那下面的信息就最后一个 microbiomics， 或者是说现在有在看肠道菌相的研究 microbiomics。那我们也听说过很多就是用 DNA 去来研究所谓的 microbiome。 觉得好，很多很高之类的。那像这样的关系，也是我们在 Omics 里面想要看的事情，跟我们生物体里面有关的各种分子，彼此之间交互作用，都是 Omics 有兴趣的对象。所以你可以想象，未来要讲这些医疗啊、健康啊，也是各式各样都要讨论。除了我们自己的细胞，我们可能要同时考虑到我们寄生的 bacteria 的这些组成。好，下来看。所以刚刚讲的就是像这个东西啊，我们有 genomics 的层级，它在意的都像 DNA 啊、RNA 变成蛋白质，这些是 genomic 的层级，还有蛋白质之间怎样被 modify， 怎样被回收，怎样降级 ，genomics 都是可以对象。Metabolomics 当然就是看这些代谢和蛋白质代谢之间这种作用，以及 phenomics 就是看我们这些前面的这种分子之间交互怎样引出来看到的表现型 phenotype。那 together 这些其实都可以算是10分biology的兴趣对象。它的终极目标当然也是整合所有的资讯，包括了我们的环境里面的 relation 导动，让我们知道我们跟环境之间交互作用，怎样影响到我们分子层级上面的所有变化。它的理想性是在这边。好，那刚刚已经说到啊，有这么多的层级，那之间的交互作用你看。那就会引领到另外一个词叫 neural medicine。好 ，neural medicine 讲的是什么？我们有很多对于系统建模的方式 ，modeling 的方式。让我们知道我们的基因，我们的刚刚提到的各种 Omics 的分子，它很合理的一个建模的方式，model 的方式就是这个 neural。各种分子之间有什么的 interaction。就用的图论的方式把它表示出来，所以用这方式我们可以建立出从分子层级的 network， 我们细胞和细胞之间的 network。比如说我们一个肿瘤的微环境，它本身肿瘤的抑制性的会让细胞跟细胞在里面，有环境和其他细胞，它们彼此之间怎样做一个交互作用？我们肠道，我们有肠道菌，还有我们的肠道的细胞啊、线条细胞，它们之间怎样做？我们的 organ 它只是之间怎样做交互作用，所以不同层级的东西我们都是可以整合在一起去看。那这个东西就是在做所谓的模板啊、medicine。那它的核心想法当然是觉得我们人体或我们的生命体系可以把它想象成 network of networks。那你从个体层级之外去看，其实网络这件事情发生在我们每天的日常生活上，像之前 Facebook 还很红的时候 ，social networks 都是一个很热门的词。我们人与人之间的关系其实都是关系嘛，我们要把它建模的话，那自然就是所谓的社交网络。那 Facebook 现在比较不红的，如果你在其他的 Instagram 之类的，其实也是类似的社交网络。所以有些做这一块的人，当然很有野心，觉得我们就要从分子层级的网络，我们个体之间的网络，social network都放开来考虑。那也有相关的医学方面的研究是在讲 ，OK， 我们social network怎样去支持、帮助我们各种 rehab 或 treatment 的效果，这样的研究也是有。那 neural medicine 这件事情，其实在很多医学院已经有一个专门特别试错去研究这一块。比如说我之前在 Harvard 的时候，工作到叫 Institute of Neural Medicine，WH 下面，那里面的老师其实都是在研究这一块，各式各样的分子器官组织之间交互作用网络。好，那我们前面已经看过 genomics， 那我觉得请讲。
Natural medicine 算 precision medicine。
好问题，那我们为什么要把它都牵扯到里面去讲？就是因为某程度上你可以把 systems biology 想象成跟 neural biology 密不可分的存在。那我们 system 要去建模， 做所谓的 modeling， 目前最普遍采用的方式就把它想象成所谓的生物网络。所以 neural medicine 或是 neural biology， 从技术面上来讲，它比较像是用数学上面图论的技术来帮助我们去了解生物，尤其是对分子的相互作用。那我们前面是有多少透露了一下，它最好用的地方是在于去研究，想看到比较高层级的这些生物分子的物质关系，这个我biology特别有帮助的地方。那 precision medicine 我们刚刚看的当然是讲很多是在 DNA 实体上面的东西，那我们也说到。比如说 NASA， 它有兴趣也不仅仅是 DNA 身上的东西，还有我们通过环境的变化，我们 stressful 的状态下面造成的一些外衣服的修饰啊，还有代谢物的改变啊。Omics 他们，也就是我刚刚说的，未来的 precision medicine 应该会往整个 Omics 的方向去看，不仅仅是现在是看genome这一部分。那既然要讲到 Omics， 就不免要提到所谓是用biology的一个方法，用biology的一个方法，所以就会扯在一起。
就是 system 听还是要 order， system 会 system 要有顺序，
顺序，
它不会有顺序，它就是可以到什么地步，一个在哪？
这也是一个很好的问题。那怎么说？你们有说过底下数据吗？那 graph 这种高中朋友听什么？我们可以用这个东西来建立模型，但这个模型可以用的比较复杂。比如说我们的 interaction association， 你可以单单的知道 OK 这两个东西有关系，比如说你跟我有关系，那我们可以进一步去 define 这个 interaction 的种类、程度，还有各式各样的 interaction， OK 把它放进来。那你刚刚说的顺序、时间性上面，就跟我们前面上一节课程到的，我们收集的 data 很好，但是对我们可能比较真正有帮助的是有时间顺序，刚刚说的。那这些东西也是可以放进来，模型里面去看。那基本上是不冲突的，尤其是在你把时间这个因素放进来的话，那在 web 上面就会有所谓的资讯流。那就会把时间这一块放进来，在什么时间上面某个在资讯层面上是讲资讯的，那都在不同时机上面可能是代谢物的反应路径，它的流量不一样，所以我们会需要一些，比如说有个方程开始要去看所谓的流量呢，时间在里面的影响。那很多当然不是像我们看到用比赛的方式去做到的。比如说，我们要看蛋白质的浓度，蛋白质的量，那我们可以去做定量，但是当我们把时间维度放进来的话，可能需要好多的时间点去做定量。然后收集到这些资讯，我们就可以知道它的流量，它变高。那蛋白质它从哪里拿来生产，它也会慢慢移位，所以这边的生产和移位的速率彼此之间的加减会相当的复杂。那这其实也是 system biology 一个很重要的东西。那你问的很好，这边没有特别讲出来，所以它牵扯到更深的东西。那你去翻一下 system biology 的课本，有很多人写的课本会特别注重在这一块，所以会用到非常多的方程。好，那我们看到Omics这边有太多字的，所以我们打算跟大家讲太深。那简单来说，Omics其实就是 DNA 做出来的这些 RNA 产物嘛。这个 RNA 产物最终会变成protein或者是哪东西？那以往都觉得只有那些变成protein的 RNA 才重要，不过后来大家都知道有各式各样的 RNA 是很重要，尽管它不是乳蛋白质，尽管它是常常跑在制造营造组织，后来都知道它慢慢有功能了。它可能会影响到其他的行业，它可能会回头来结合到我们的DNA 的产物，去做一些调控动作。好，那这边我们就跳过了。 传统的生物学上面做的实验，或许可以对一个分子里面的一个点看得非常清楚。但是当我们收集到够多的东西，或许比较能看得清楚后面确什么是这样子。OK， 所以这是我们喜欢说出来的。好，那要做到这件事情的话，其实我们就需要一些高通量的仪器。就像刚刚说的，我们已经提到地区，我们需要高通量的地区。高通量的意思是什么？我们不是从头读到尾，然后开始，所以它的策略是大概是shotgun，把 DNA 打碎，在一个物品上面去同时进去传播的去放。那 DNA microarray 其实是比较前面的技术，到现在还是用蛮多的。不过在市场上它慢慢飞到。那它做的是什么？你定序里出来，高通量定序里是出来之前，很大部分我们需要依赖 DNA microarray。那我觉得我们也没有时间去看它的细节。我觉得我们大概讲一下好了，它就是一个所谓的晶片的意思。所以以前你会听到我们什么 DNA 晶片、DNA 晶片，其实利用的这种东西，它结合了我们半导体微流体的技术。小小的一块晶片，它上面有非常非常多，像刚刚我们已经做好的探针 DNA 探针，我们已经知道 DNA 序列探针会把它结合在上面。那我们接下来要做的是，我们取一个 sample， 比如说抽血，你抽出你的血液里面的某些血细胞的 DNA， 很明显知道的。那它的 DNA 你就可以长交到上面。那有结合到的部分就会结合到，剩下就死掉，那就知道哪些序列是有长交到的。那就可以知道上面到底有哪些 DNA 是留在上面的。这边它有说到，就像这样。所以这是一个比较早期的技术。那左右两边是这个技术的两种分支。那我们合成的 DNA， 我们知道我们可以做出这种载体。上面有各式各样的探针。那我们再把我们的样本收到了，比如说这个是软胚，我们去用3、3、5去做 labeling， 所以有不同的重量度去做 hybridization， 那就会看到这样的一个玻片上面有不同强度的颜色，可能有比较绿的、比较红的，那你就可以知道来自这个样本的某些样本比较强。是来自这个样本的东西比较强。那这就是为什么前面看我们那边什么长，以及长出来这个影响。那右边是另外一种方式，右边就刚刚看到的 Omics 那支片，它用半导体技术去长，它就从我们的一样是类似载玻片一样的东西去长出来我们想要的探针。所以他们讲的是4:4的4:4，它接着上去长。不像左边是用推理的方式，这比较像通过表记，这个比较像通过表记。那右边其实也是一样，我们实验就分成 K3 control， 分别去做它的DNA去做杂交，一样去侦测它的梦想，就可以去比较出来我们的 K3 control在各式各样的基因上面，哪些基因的表现出现差异。这个是它的大意。那我觉得我们可以跳过这一块，那直接看到最后。那我之所以要讲这个东西，是因为我们接下来，因为我看大部分只能来自脑科出来，所以我想我就提一个 real world example 一个例子。那这个例子是 Alan Jones， 不知道在你们那领域是不是相关的？在我们这个领域，他算是有名，因为他有一个 Alan Institute， 他专门收集一些好组织的基因检测的结果，那也就是他这个 talk 里面要讲的，他介绍一下他们的工作。那你会发现这个 talk 已经大概8、9年前了。那他们收集出来的资源，目前是在脑组织基因表现 Omics 的研究上面很重要的资源。所以我觉得可以利用今天这个机会，特别是刚刚大家已经知道什么是 Omics， 什么是 DNA microarray。之后我们可以看一下它到底在做什么东西。那15分钟我们看完应该刚好下课，这是我们最后一个内容，大家看一下实际上的例子。好，
long-held fascination for the human brain. We chart it, we've described it, we've drawn it, we've mapped it. Now, just like the physical maps of our world that have been highly influenced by technology, think Google Maps, think GPS, the same thing is happening for brain mapping, true transformation. So let's take a look at the brain. Most people, when they first look at a fresh human brain, they say it doesn't look like what you're typically looking at when someone shows you a brain. Typically what you're looking at is a fixed brain, it's gray, and this outer layer, this is the vasculature, which is incredible around the human brain. This is the blood vessels. 20% of the oxygen coming from your lungs, 20% of the blood comes from your heart, is surfacing this one organ. That's basically, if you hold two fists together, it's just slightly larger than the two fists. Scientists sort of in the end of the 20th century learned that they could track blood flow to map non-invasively where activity was going on in the human brain. So for example, they can see in the back part of the brain, which is just turned around there, there's the cerebellum, that's keeping you upright right now, it's keeping you standing, it's involved in coordinated movement. On the side here, this is the temporal cortex. This is the area where primary auditory processing, so you're hearing my words, you're sending it up into higher language processing centers. Towards the front of the brain is the place in which all of the sort of more complex thought, decision-making is the last to mature sort of late adulthood. This is where all your decision-making processes are going on. It's the place where you're deciding right now you probably aren't going to order the steak for dinner. So if you take a deeper look at the brain, one of the things if you look at it in cross-section, what you can see is that you can't really see a whole lot of structure there. But there's actually a lot of structure there. It's cells and it's wires all wired together. So about 100 years ago, some scientists invented a stain that would stain cells. And that's shown here. In the very light blue, you can see areas where neuronal cell bodies are being stained. And what you can see is this very non-uniform. You see a lot more structure there. So the outer part of that brain is the neocortex. It's one sort of continuous processing unit, if you will. But you can also see things underneath there as well. And all of these blank areas are the areas in which the wires are running through. They're probably less cell dense. So there's about 86 billion neurons in our brain. And as you can see, they're very non-uniformly distributed. And how they're distributed really contributes to their underlying function. And of course, as I mentioned before, since we can now start to map brain function, we can start to tie these into the individual cells. So let's take a deeper look. Let's look at neurons. So as I mentioned, there are 86 billion neurons. There are also these smaller cells, as you'll see. These are support cells, astrocytes, glia. And the nerves themselves are the ones who are receiving input, they're storing it, they're processing it. Each neuron is connected via synapses to up to 10,000 other neurons in your brain. And each neuron itself is largely unique. The unique character of both individual neurons and neurons within a collection of the brain are driven by fundamental properties of their underlying biochemistry. These are proteins. They're proteins that are controlling things like ion channel movement. They're controlling who nervous system cells partner up with. And they're controlling basically everything that the nervous system has to do. So if we zoom in to even deeper level, all of those proteins are encoded by our genomes. We each have 23 pairs of chromosomes. We get one from mom, one from dad, and on these chromosomes are roughly 25,000 genes. They're encoded in the DNA, and the nature of a given cell driving its underlying biochemistry is dictated by which of these 25,000 genes are turned on and at what level they're turned on. And so our project is seeking to look at this readout, understanding which of these 25,000 genes is turned on. So in order to undertake such a project, we obviously need brains. So we sent our lab technician out. We were seeking normal human brains. What we actually start with is a medical examiner's office. This is a place where the dead are brought in. We are seeking normal human brains. There's a lot of criteria by which we're selecting these brains. We want to make sure that we have a normal human between the ages of 20 to 60. They died a somewhat natural death with no injury to the brain, no history of psychiatric disease, no drugs on board. We do a toxicology workup. And we're very careful about the brains that we do take. We're also selecting for brains in which we can get the tissue, we can get consent to take the tissue within 24 hours of the time of death. Because what we're trying to measure, the RNA, which is the readout from our genes, is a very late while, so we have to move very quickly. One side note on the collection of brains, because of the way that we collect and because we are requiring consent, we actually have a lot more male brains than female brains. Males are much more likely to die accidental death in the prime of their life, and men are much more likely to have their significant other spouse give consent than the other way around. So the first thing that we do at the site of collection is we collect what's called an MR. This is magnetic resonance imaging, MRI. It's a standard template by which we're going to hang the rest of this data. So we collect this MR, and you can think of this as our satellite view for our map. The next thing we do is we collect what's called the diffusion tensor imaging. This maps the large cabling in the brain. And again, you can think of this as almost mapping our interstate highways, if you will. The brain is removed from the skull, and then it's sliced into 1 centimeter slices. And those are frozen solid, and they're shipped to Seattle. And in Seattle, we take these, this is a whole human hemisphere, and we put them into what's basically a glorified meat slicer. There's a blade here that's going to cut across the section of the tissue and transfer it to a microscope slide. We're going to then apply one of those stains to it, and we scan it. And then what we get is our first mapping. So this is where our experts come in and they make basic anatomic assignments. You could consider this state boundaries, if you will. It's pretty broad outlines. From this, we're able to then fragment that brain into further pieces, which then we can put on a smaller cryostat. And this is just showing us here, this frozen tissue, and it's being cut. This is 20 microns thin, so this is about a baby hair's width. And remember, it's frozen, and so you can see here old-fashioned technology with a paintbrush being applied. We take a microscope slide, and we very carefully melt onto this slide. This will then go to a robot that's going to apply one of those stains to it. And our anatomists are going to go in and take a deeper look at this. Again, this is what they can see under the microscope. You can see collections and configurations of large and small cells in clusters in various places. And from their expertise, they understand where to make these assignments, and they can make basically what's a reference atlas. This is a more detailed map. Our scientists then use this to go back to another piece of that tissue and do what's called laser scanning microdissection. So the technician takes the instructions, they scribe along a place there, and then the laser actually cuts. You can see the blue dot there cutting, and that tissue falls off. You can see on the microscope slide here, that's what's happening in real time. There's a container underneath that's collecting that tissue. We take that tissue, we purify the RNA out of it using some basic technology, and then we put a fluorescent tag on it. We take that tagged material and we put it onto something called a microarray. Now, this may look like a bunch of dots to you, but each one of these individual dots is actually a unique piece of the human genome that we spotted down on glass. This has roughly 60,000 elements on it, so we repeatedly measure various genes of the 25,000 genes in the genome. And when we take a sample and we hybridize into it, we get a unique fingerprint, if you will, quantitatively of what genes are turned on in that sample. Now we do this over and over again. This process for any given brain, we're taking over 1,000 samples for each brain. This area shown here is an area called the hippocampus. It's involved in learning and memory, and it contributes to about 70 samples of those 1,000 samples. So each sample gives us about 50,000 data points with repeat measurements, 1,000 samples. So roughly we have 50 million data points for a given human brain. We've done, right now, 2 human brains worth of data. We've put all of that together into one thing. I'll show you what that synthesis looks like. It's basically a large data set of information that's all freely available to any scientist around the world. You have to log in to come use this tool, mine this data, find interesting things out with this. So here's the modalities that we put together. You'll start to recognize these things from what we've collected before. Here's the MR. It provides the framework. There's an operator side up on the right that allows you to turn. It allows you to zoom in. It allows you to highlight individual structures. But most importantly, we're now mapping into this anatomic framework, which is a common framework for people to understand, where genes are turned on. So that red levels are where a gene is turned on to a great degree. Green is those sort of cool areas where it's not turned on. And each gene gives us a fingerprint. And remember that we've assayed all the 25,000 genes in the genome and have all of that data available. So what can scientists learn about this data? And we're just starting to look at this data ourselves. There's some basic things that you would want to understand. Two great examples are drugs, Prozac and Wellbutrin. These are commonly prescribed antidepressants. Now remember, we're assaying genes. Genes send the instructions to make proteins. Proteins are targets for drugs. The drugs bind to proteins and you either turn them off, etc. So if you want to understand the action of drugs, you want to understand how they're acting in the ways you want them to, and also in the ways you don't want them to, in the side effect profile, etc. You want to see where those genes are turned on. And for the first time, we can actually do that. We can do that in multiple individuals since we've assayed two. So now we can look throughout the brain, we can see this unique fingerprint, and we get confirmation that indeed the genes turned on for something like Prozac in serotonergic structures, things that are already there. But we get to see the whole thing. We also get to see areas that no one has ever looked at before, and we see these genes turned on there. This is interesting side effects it could be. One other thing you can do with such a thing is you can, because it's a pattern matching exercise, because there's a unique fingerprint, we can actually scan through the entire genome and find other proteins that show a similar fingerprint. So if you're in drug discovery, for example, you can go through an entire listing of what the genome has on offer to find perhaps better drug targets and optimize. Most of you are probably familiar with genome-wide association studies in the form of people covering the news saying scientists have recently discovered the gene or genes which affect X. And so these kinds of studies are routinely published by scientists, and they're great. They analyze large populations, they look at their entire genomes, and they try to find hotspots of activity that are linked causally to genes. But what you get out of such an exercise is simply a list of genes. It tells you the what, but it doesn't tell you the where. And so it's very important for those researchers that we've created this resource. Now they can come in and they can start to get clues about activity. They can start to look at common pathways, other things that they simply haven't been able to do before. So I think this audience in particular can understand the importance of individuality. And I think every human, we all have different genetic backgrounds, we all have lived separate lives, but the fact is our genomes are greater than 99% similar. We're very, very similar at the genetic level. And what we're finding is actually, even at the brain biochemical level, we are quite similar. And so this shows it's not 99%, but it's roughly 90% correspondence at a reasonable cutoff. So everything in the cloud is sort of roughly correlated. And then we find some outliers, some things that lie beyond the cloud. And those genes are interesting, but they're very subtle. So I think it's just an important message to take home today that even though we celebrate all of our differences, we are quite similar even at the brain level. And what do those differences look like? This is an example of a study that we did to follow up and see what exactly those differences were. And they're quite subtle. These are things where genes are turned on in an individual cell type. These are two genes that we found as good examples. One is called relin. It's involved in early developmental cues. DISC1 is a gene that's deleted in schizophrenia. These aren't schizophrenic individuals, but they do show some population variation. And so what you're looking at here in donor 1 and donor 4, which are the exceptions to the other two, that genes are being turned on in a very specific subset of cells. It's this dark purple precipitate within a cell that's telling us a gene is turned on there. Whether or not that's due to the individual's genetic background or their experiences, we don't know. Those kinds of studies require much larger populations. So I'm going to leave you with a final note about the complexity of the brain and how much more we have to go. I think these resources are incredibly valuable. They give researchers a handle on where to go, but we've only looked at a handful of individuals at this point. We're certainly going to be looking at more. I'll just close by saying that the tools are there, and this is truly an unexplored, undiscovered continent. This is the new frontier, if you will. And so for those who are undaunted but humbled by the complexity of the brain, the future awaits.
好的，是的。那我们今天大家。 条线。OK， 好，那今天就到这边了，谢谢大家。